later? Okay. Okay, thank you, Lisa, for a great talk. Next, I'd like to introduce Professor Aydogan Ozcan from UCLA. He's also HHMI investigator, and at his young age already has gathered too many laurels to list, I have to say, so I'm I'm not going to start, other than to say he's a real-life inventor. He has a huge amount of patents already and works in the bioengineering field. And he's going to talk to us about mobile imaging and sensing technologies for diagnostics. He's also received a Presidential Early Career Award for Scientists and Engineers and is a member of the National Academy of Inventors. So welcome, Aydogan. And we'll try to make the screen. Yes.
Awesome.
Perfect.
Great.
If you need it lighter, you just go on presentation.
Okay, great. Thank you so much. All right. Well, good morning, everyone. Thanks for having me here. It's a great pleasure to be here and share with you some of our work on the use of computation and optics to create new types of imaging and sensing systems with applications in medical diagnostics. So in a nutshell, my lab is aiming to democratize measurement tools, advanced measurement tools. And in this slide, I'm sharing with you some examples of how we've been actually utilizing computation and optics to create mobile, cost-effective interfaces to conduct advanced microscopic imaging of specimen and sensing. So this is these are the results of actually a long journey. And when we first started to play with mobile phones as a platform, we could barely see red blood cells. But now, the smartphone itself can be used as a platform for advanced microscopic imaging, bright field or fluorescence imaging of specimen. It's really come a long way from barely seeing a red blood cell. Now you can see individually labeled viral particles, even DNA molecules can be seen with very inexpensive attachments to smartphones, converting them into microscopes. For example, in this work, you're looking at a fluorescence microscope that's specifically aiming to analyze DNA with a very inexpensive and simple sample preparation, which essentially stretches DNA molecules put on a glass slide that are labeled with intercalating dyes. We can see individual double-stranded DNA molecules and size them with about 1 kilobase pair sizing accuracy, nicely matching to a benched-up standard fluorescence microscope. As shown here, looking at the same field of view. You can create multimodal microscopy tools using the smartphone as a platform. For example, here I'm sharing with you 2 fluorescent channels installed on the same smartphone in addition to bright field microscopy. So bright field microscopy and 2 different channels of fluorescence can be superimposed. In this example, we're looking into reading of rolling circle amplification products, RCPs, for in situ mutation analysis conducted in colon cancer patients. And what you see here are 2 different colors that label the RCA products in 2 different fluorescent tags, colors, revealing at the cellular level essentially wild type versus mutant for essentially next generation DNA sequencing for diagnostics. So as I mentioned, it's really a long way since we started to play with smartphones for microscopy and sensing. In addition to microscopy, we're also very much interested in using the smartphone as a sensor of various different kinds. Some earlier work, we've used it to detect waterborne pathogens. For example, here is a device that is a fluorescence microscope, but it has a front end to screen Giardia cysts. Where you have a porous membrane here, you put your sample, several milliliters of water. Because of the pore size matched to the size of the Giardia cyst, they're stuck there and they're also fluorescently tagged. Without a need for a microbiologist, you can actually achieve about a cyst per mil level of detection sensitivity using fluorescence microscopy, this front-end disposable cartridge, and machine learning at a simple server, which could be your local laptop as well. Some other examples are shown here. This is also my conflicts of interest disclosure that I have 2 startup companies commercializing some of these technologies, and some of these work actually has resulted in products being used, such as this one, for example, which is a fluorescent, chemiluminescent, and colorimetric diagnostic test reader for various different lateral flow tests or vertical flow tests. This one is another very interesting device, which is actually a plate reader for various different applications for ELISA tests or other types of turbidity assays. You can utilize this smartphone-based reader for quantifying a standard 96-well plate in an easy, high-throughput manner. Here's how it looks inside. Basically, a standard plate reader in your labs, which might be 10,000, 20,000, depending on what kind of features you have, would be having a scanning optical system to read all these wells one by one. This is a snapshot imager. Thanks to these plastic, inexpensive fiber optic cables, we're mapping the entire 96-well plate into a tiny fiber bundle which is imaged. And we're quantifying whether it's the turbidity, transmittance, colorimetric ELISA or fluorometric ELISA in a different design. We can actually read them in a snapshot without any scanning systems. Very lightweight and handheld system. We've tested it with actually patient samples against FDA-approved test readers. In one study, we used about 1,100 patient samples for IgG assays for mumps, measles, HSV1, and HSV2 for vaccination testing. And we've achieved extremely well performance matching the results of an FDA-approved device in terms of specificity, sensitivity, and agreement. Processing those transmission images of those fiber bundles and some machine learning geared toward each one of these tests, we've achieved almost FDA-ready data without any very major or major errors made for diagnostic purposes. So it's indeed not only mobile, cost-effective, but also has the analytical sensitivity and performance that is coming very close to FDA-approved devices, as we've shown in this study. And in a subsequent work, we've utilized the same platform for looking into antimicrobial resistance. This is an MIC measurement, minimum inhibitory concentration measurement at UCLA Health. Basically, an MS-level technician is looking at these 96-well plates, which contains a patient isolate, and it has a panel of antibiotics. In this case, we have about 19 different antibiotics at different concentrations. And the goal of this technician is to prescribe the minimum inhibitory concentration for that patient isolate. This is today done manually because, believe it or not, that's the gold standard. That's beating out anything else. This is not recognition of the organism. This is essentially organism isolate that is being tested for MIC measurements. We've shown also a very good match to an expert diagnostician for these MIC measurements performed on patient isolates, showing that it's actually coming a long way in terms of its performance in addition to its mobility and cost effectiveness. So some of these devices are using the smartphone as a platform for microscopy and sensing at the intersection of computation and optics. At the same time, sometimes we're very interested in utilizing components from the smartphone to form computational images. In fact, what you see here on the left, these are computational microscopes where the main component, in addition to computation, is a Cmos imager, the same 10 megapixel, 20 megapixel imager of a smartphone. And this has come a long way in terms of microscopy and in terms of sensing. If you look at one of those devices, actually it's very inexpensive and simple to put together. You have a Cmos imager taken off a smartphone. This is literally the same Cmos imager that many of your smartphones' camera interfaces have. We start with 5 megapixel images. Nowadays, we have maybe 20, 30 megapixel imagers. It's the same platform. The sample goes from the site. It's a transmissive sample. There are some LED lighting here. These are all automatically controlled, which means there is nothing manual here. As the sample goes in, these light sources are one by one turned on and off, giving us enough degrees of freedom to synthesize an image that is almost diffraction limited, taking those relatively large pixel sizes of Cmos images, 1 to 2 micron these days, and creating sub-pixel, effectively sub-pixel images that can reach to a resolution of sub 0.5 micron if you actually do it right with the right reconstruction algorithms, which utilize nowadays deep learning and machine learning combination. So just a comparison, it's a very lightweight interface. as tall as your iPhone, maybe 200 grams, very inexpensive to put together because of the fact that it's benefiting from economies of scale. Cmos imagers obviously are a few dollars because of the large volume of our smartphones. And the rest is just LEDs and simple fiber optic cables put together. These are some earlier images of how this kind of a microscope is working. This is a very early generation CMOS imager, 5 megapixel. This is a thin smear. This is how it looks under a standard bright field microscope. These are signatures of Plasmodium falciparum. This is how it looks in the phase channel of this computational microscope. One advantage of this platform, in addition to being field portable and inexpensive and lightweight, is that it can look at a very large area. This is, for example, a larger section of that thin smear, and if you zoom in, you'll be able to see zoomed in images of these red blood cells that are infected with parasites. So we have about 2 orders of magnitude wider field of view, which is better highlighted here. This is a thin smear. A typical 40x objective lens would be looking at this circular region of interest. That's why you wouldn't need to normally scan the sample and capture multiple images. The lensless imaging system essentially has a field of view that is sometimes 20, sometimes 30 millimeters square, depending on what kind of a CMOS imager you use. And you can bring color imaging to this kind of a system based on multicolor illumination. These are some recent results from the state-of-the-art for lensless imaging. This is an H&E stained lung pneumonia sample. It's about a few microns in thickness, standard histochemical staining process. It's also highlighting the field of view comparison here with different objective lenses. 20x is this green region. You can take a region of interest digitally and zoom in and zoom in and continue zooming in. You have there, as I said, almost diffraction limited resolution, which means effectively the pixel size here can be as small as about 200 nanometers, even with using CMOS images that have 1 to 2 micron pixel size. This is a framework called pixel super resolution that's giving us, from let's say a 10 megapixel imager, 20 megapixel imager, approximately 1 billion useful pixels across a very large field of view. So in addition to all the work that we've done on pushing the resolution of this system, we've also pushed the sensitivity of this system so that it can be used as a sensor. In this work that I'm showing here, we're utilizing the same lensless chip-scale computational microscopy platform for looking into individual viruses and nanoparticles. They're tiny. These are 100, 200 nanometer particles, sub-wavelength. That's why it's very difficult to detect them with a bare imaging system. In fact, before we play some tricks, we cannot detect them. It's noise floor as shown here. The trick here is to use essentially some condensation of polyethylene glycol. We heat it up to about 100 Celsius. The vapor is condensing. By the time it reaches the sample here, the nanoparticles and viruses are facing down. The vapor condenses in a matter of essentially a few minutes, forming around each nanoparticle or virus of interest tiny nano lenses, which increases the refractive power of that nano lens and helps us to detect them after condensation. The smallest particle size that we can these days detect is around 30 to 40 nanometers. This is an entirely controlled process. We can actually, we know exactly when it's diminishing returns and we stop the entire image acquisition. Depending on how fast the evaporation is, after maybe a minute, you've reached the maximum sensitivity. That's when you stop the entire process. And this has also, it's not just a yes or no decision. It's not just detection. It's actually quantification of the size as well. Based on the phase signature, which is quantified in terms of optical path length, we can have a sizing accuracy of these particles about 11 nanometers, all the way from small particles like 30, 40 nanometers to several hundred nanometers and above a micron can be imaged and sized with this platform. It's a detection system, but with surface chemistry, it can also serve as a sensor. We've functionalized substrates, glass-based substrates, to specifically capture a certain viral particle, in this case HSV, to use the specificity of the surface chemistry to isolate HSV particles from a sample, and at the same time use the sizing capability and detection capability of this lensless microscope to have an additional degree of specificity based on size to reject non-specifically bound particles that are smaller or bigger than the expected HSV size. This has achieved a very nice detection limit of a few viral particles per liquid sample that you put in there, which can be a few microliters in volume. And because it's a platform, you can actually use this handheld system to read out various different types of sensors. Literally, it's a platform. It can be used for many things. In one recent work, we've used this for bead clustering-based assays. Basically, there are microscale particles, a few microns in diameter, that are functionalized to be specific to a certain viral particle. In the presence of the viral particle, they cluster. This is sometimes read with visual inspection of a technician. Here we're using the chip scale lensless microscope to quantify these binding events, which essentially correlate with the concentration of the viral particle. If there is no viral particle, we get very few clusters in your sample. As you start to increase the copy number of viral particles per microliter, you start to increase the number of clusters in your sample. We've achieved about 5 viral copies per microliter sensitivity with this kind of a platform, just as a readout based on also machine learning in the way that it's processing these holograms, these diffraction patterns captured by an inexpensive imaging system. So these are giving you some examples of how this computational platform with essentially a Cmos Imager, literally that's being used in our smartphones, can perform advanced microscopic imaging over a large sample area, and at the same time use it for sensing purposes. So it's a nice platform that I think can be used for various different diagnostic purposes. In my last part of the talk, I'd like to tell you how we've actually taken the same framework and used it as a sensor for motility. So everything that I've shown you so far, they were actually static samples. They were static on a substrate captured specifically or non-specifically, sometimes tissue samples. So everything was static. And I could actually change the illumination the way that I like to enhance the resolution, contrast, etc. So now I'd like to switch to a case where I'd like to use motility, locomotion, as a biomarker, and still be able to use this inexpensive portable system or the principle of imaging and sensing to create some unique form of diagnostic tools. Here we're looking at actually a label-free and high-throughput detection platform for specifically looking at large volumes, a few milliliters of turbid media, like, for example, whole blood. And in this case, because it's label-free, our sensitivity will come from motility of the biomarker. In this case, we're looking at various different trypanosomes, and these are essentially affecting hundreds of millions of people. And some examples are shown here. The same is true for Trichomonas vaginalis, which is essentially the #1 non-viral STD. So motility is critical for infection. The power of infection of these parasites is literally powered by their ability to move. And we'll now use that as a specific biomarker to be able to isolate the signatures of these parasites within whole blood samples or within large volumes of bodily fluids. This is a very difficult problem because typical parasitemia for trypanosomes, for example, is less than 100 per mil. If you look at a standard optical microscope, let's say 20x objective that you use, the volume that it looks at is about a nanoliter, let's say a few nanoliters. There's a huge mismatch here in terms of throughput. So that means it's like finding a needle in a haystack. We need technologies that are high throughput to be able to screen large volumes without the tools to separate these or concentrate these parasites. That's exactly our goal here, with minimal sample preparation, without the need for an expertise, quick sample preparation and rapid screening of large volumes. The underlying principle here will be speckle analysis. If you're looking whole blood and try to shine light through it, all you get is speckle because there are billions of other cells in there. And your parasite there is essentially just one of those. And it's very difficult to see it. But if you look at the speckle patterns as a function of time and how they're changing, you can actually spot these tiny parasites within billions of other cells around it. The advantage of the platform that I'm going to show you is it's high throughput, label-free, minimal sample preparation, maybe 3 minutes, and it can screen in automated computation and everything with a local laptop about 3.2 milliliters with a decision, final decision given in about 20 minutes, including the sample preparation. And this requires no, essentially, expertise. Here's the front end, which is 3 minutes. You have a lysis buffer to explode the red blood cells mostly. And then you mix the sample. So this is actually a few seconds, as you can see. The most important part here is to fill these rectangular capillaries. Each one of these is taking about 1 milliliters of whole blood. So this is essentially being screened by a device that I'm going to show you. So this sample preparation, most of it is essentially filling these capillary tubes. These are disposable tubes that are going to be imaged with a device that we created. The scanning time after all that 3 minutes of sample preparation is taking about 11 minutes with a device that has 3 different heads, each of which is looking at one of these capillaries. There's nothing magical about 3, but we're benefiting here from our cost effectiveness and compactness so that in a lightweight device, about 1.6 kilograms total, you can actually have 3 of these in a cost-effective manner being controlled in parallel. Here's a movie of how it's working. This is a movie that is showing you 16x speeded, showing how it's essentially performing. These are 3 individual samples. The head is scanning and capturing high frame rate, about 30 frames per second for every single spot. For every single spot, it's looking at if there's locomotion there by analyzing those, let's say, 60 frames taken in about 2 seconds. It's randomly moving, hopping from different positions, not to heat up the sample. So we've done some analysis that the illumination over that few seconds might heat up and cause some gradient of heat. As a result, we might have false detection because RBCs, red blood cells, and other debris might essentially be moving. So that's why the locomotion of this head is designed in a smart way to not heat up the sample. So everything that is moving there is really the true trypanosomes. And after the acquisition of this, in fact, during this entire 11 minutes, a laptop is processing these images as they come, which means it's doing the parasite detection. Raw images look like speckle patterns. You cannot really spot anything here. But again, it's the differential analysis over those 50, 60 frames within that 2-second window is what we analyze at the end to give you a contrast that correlates with motility. And that motility is the trypanosome signature within that 1 milliliter of rectangular tube. In these results that I'm showing, we use the BSL-2, a non-human infectious subspecies of trypanosoma. So we diluted that in whole blood samples to get a titration curve for understanding the sensitivity of this system. This is how it looks after the motility analysis. I'll show you just 3 trypanosomes. These are raw images. Because of whole blood, there's a lot of speckle, very hard to see. The phase signature, if you carefully look at the phase signatures, you can actually see the wiggling of the live parasite there. That's essentially the signature that's boosted up after differential analysis to these bright spots over there. So this is happening across all those 3 tubes in parallel to screen about 3.2 milliliters in about a computation time of a few minutes with a laptop. So this is our detection results as a function of spiked trypanosome concentration. We have about 10 parasites per mil in whole blood level of detection limit, which is compared to other techniques, which is slightly better by about 5x. And in fact, considering the fact that the front-end sample preparation is only 3 minutes, we have a huge advantage here because you don't need expertise to put the samples in, and the analysis is done entirely automated. This detection limit improves with spinal fluid samples. This is actually a stage 2 of sleeping sickness. At that stage, the parasite starts to attack the central nervous system, and you need to change the treatment at that stage. That's why looking at CSF is also quite important. For CSF samples that are spiked with trypanosomes, we were able to achieve about 3 trypanosomes per mil which is mostly because of the fact that CSF is much more transparent and we don't have the debris and other things moving from the background of whole blood. That's why it's better. And the movie here will show you also that the signature of the trypanosome in CSF buffer is also much more clear. Look at, for example, this phase row of image. You can clearly see the signatures of the parasites, the wiggling. And that's essentially differentially analyzed to give you these very high contrast and signal-to-noise ratio spots that reveal that trypanosome. The same platform can be applied to any other motile parasite that you would like to detect in large volumes. In this case, we're looking at Trichomonas vaginalis, which is the cause for the #1 non-viral sexually transmitted disease. As you can see, these are cultured parasites in a buffer solution. They have a huge contrast because they're relatively larger in this case, and in the phase signature, you can clearly see them. So just to highlight, you can have a platform using this technology for various different motile parasites to be screened rapidly in high throughput. To sum up, this is a unique technology in the sense that it's label-free. It's using locomotion as a biomarker and holographic speckle patterns to look at large volumes on the order of 3.2 milliliters in this case. The entire sample preparation starts with 3 minutes of very minimal sample prep, most of which, about 2 minutes of which actually is sample loading. With automated systems, these capillaries can be actually loaded much faster so that this can be almost maybe a few seconds in next generation devices. And the rest is just imaging plus processing done total under about 17 minutes. A good detection limit has been achieved in whole blood, about 10 trypanosomes per mil. In CSF samples that are spiked with trypanosomes, it's 3. So this current research prototype that I've shown you is about $1,850. That's essentially a single unit. We believe with large volume manufacturing that can go under easily $500 with these components purchased in large quantities and optimized for cost. So it's a platform which we're very excited about, and hopefully this summer would like to start testing this with some of our colleagues in a developing country in Africa. So to summarize and conclude, computation and optics intersection provides us some very unique opportunities to democratize advanced measurement tools. And I'll give you some examples of that from our lab. For microscopy, the use of microscopic imaging for various different biomedical samples of interest for diagnostics. At the same time, these tools are helping us to be used as sensors, sensory instrumentation, for various different platforms, including 96-well plates or other types of cartridges that are custom designed for the sensing diagnostic test at hand. With this, I'd like to conclude and acknowledge our funding agencies and all the lab members behind this work. And thanks again for your attention and invitation.